session chair, that is Dr. V.K. Dikshit, and our keynote speaker, Dr. Prabhat Arya, to our esteemed audience. I would like to invite Dr. V.K. Dikshit on stage. Now, I would request Dr. Rizwan Khan, Principal Skyper, to greet Dr. V.K. Dikshit with a bouquet. Professor V.K. Dikshit was inducted in pharmacy profession when he joined as B.Pharm student at Department of Pharmaceutical Sciences and earned B.Pharm degree in 1968. Later he joined M.Pharm program at the same department with specialization in pharmacognosy and passed M.Pharm in 1970. After doing his M.Pharm, he pursued PhD program under the supervision of Professor K.C. Verma. Under the award of UGC Senior Research Fellowship, PhD degree of University of Sagar was awarded to him in 1974. In 1975, he joined as lecturer in Department of Pharmaceutical Sciences at University of Sagar. In 1981, he was selected as reader, and in 1986, he became professor of pharmacognosy and phytochemistry. As lecturer and reader, he was assigned teaching of pharmaceutical engineering, which imparted the flavor of the pharmaceutical in his teaching and research. Besides his primary interest in pharmacognosy, he retired from active university services in November 2012 and worked as Dean Academics at Bhagyodaya Teerth Pharmacy College, Sagar, for 2 years. Now, I would request Dr. V.K. Dikshit to throw the light on the topic.
The topic of this session lecture is understanding and tackling undruggable territory in the drug discovery arena, and the speaker is Dr. Prabhat Arya. Dr. Arya is a co-founder and chairman, Smerian Inc. He is also a co-founder of Stealth Mode Biotech. He has also worked as distinguished research professor in various reputed universities. He has spent more than 20 years of this academic career at National Research Council of Canada, and also has a brief stint with Ontario Institute for Cancer Research. He is a champion of building integrated research teams for tackling complex biological questions that are not possible to achieve by individual efforts. He is now full-time committed to setting up biotech path for harnessing his academic research. He has published more than 105 publications and 170 invited guest lectures. He has also trained 55 postdoctoral fellows and has 12 PhD students. Dr. Arya is passionate about connecting academic science research with the real business world and thoroughly enjoys sharing his experience with the younger generation. With such a renowned and gifted speaker, I am sure the topic that he has selected, that is undruggable territory, will be well covered. I invite Dr. Arya for his lecture. Dr. Arya, please.
Due to some technical glitch, we are going. We are moving to our next session. Now, I would like to introduce you all with our next chair, Dr. S.K. Jain, and our keynote speaker, Dr. Frederick E. Williams, to our esteemed audience. I would like to invite Dr. S.K. Jain on the stage. And Dr. Williams. Now, I would request Dr. Piyush Trivedi sir to greet Dr. S.K. Jain with a bouquet. Williams sir also. Dr. S.K. Jain. Dr. Sanjay K. Jain is a professor of pharmaceutics presently working in the Department of Pharmaceutical Sciences. Dr. Hari Singh Gaur, Vishwavidyalaya, Sagar, MP. At present, he is also holding position of Director, Planning and Resource Generation of the University Chairman Council for Industry Higher Education Collaboration and Chairman of University IPR Cell. He is former head of Department of Pharmaceutical Sciences and also former Dean of School of Engineering and Technology at Dr. H.S. Gaur Vishwavidyalaya, Sagar, Madhya Pradesh. With over 33 years of experience in teaching and research and 2 years in industry, he has 197 research publications with more than 52 H index and 150 I-10 with over 10,000 citation index. He has also written a book and more than 40 book chapters in different national and foreign reference books. He has 1 US and 1 European patent to his credit and also filed 3 Indian patents. His main research area is design and development of various new drug delivery systems for the treatment of disease associated with gastrointestinal tract and brain. Professor Sanjay has maintained high standard of research and teaching throughout his service to the university. Now I would request Dr. S.K. Jain to throw the light on the topic for and invite Dr. Frederick E. William on the stage.
First of all, I would like to thank Professor Piyush Trivedi ji for giving me a chance to chair a session of a renowned scientist, Dr. Frederick E. William. I'm also thanking to the organizer for giving me such a nice platform. Today you will be having a renowned scientist who is working on the zebrafish, and he will be delivering a lecture on the modeling zebrafish as a research tool, scope and practices. Dr. Frederick is a professor, a chairperson of Department of Pharmacology and Experimental Therapeutics. He has pursued his PhD in medical science and biochemistry from Medical College of Ohio in 1990. He has completed his postdoctoral in medicinal chemistry from University of Toledo from 1996 to 200, and also in biochemistry from Medical College of Ohio in 1992 to 96, as well in chemistry from University of North Carolina from 1990 to 92. Dr. William has helped organized and participated in ECAT, an international meeting held at University of Toledo Health Science Campus in 2017. Currently, he is working on the development and behavior toxicity of drugs and environmental chemical, and also on animal model of human diseases and disorder using zebrafish. His recent article was published in 2020 about combining neurobehavioral analysis and in vivo photoaffinity labeling to understand protein targets of methamphetamine in Casper zebrafish, and he have many more published articles in some reputed journals. He is the member of many learned society. So with this brief I invite Dr. Frederick for his deliberation. He will be delivering on modeling zebrafish as a research tool, scope and practices. Dr. Frederick, please.
I want to say I am very thrilled to be here, and we're looking forward to interacting with the people here due to the MOU that was just signed. What I want to What I want to do with this lecture is take you through drug development, which usually is drugs being developed as a chemist, putting them in through cell culture, etc., to try and come up with some very interesting pharmacological, toxicological properties, and then going straight usually into mice or rats to see if there's an animal model that would be involved. In my case, I want to convince you that zebrafish belong there as well. That zebrafish can be an animal model that can be between our cell culture and our rodents. So my talk here will talk about zebrafish, use of zebrafish in that way. I am from the University of Toledo in Toledo, Ohio, and my colleague, Dr. Tillore, actually talked about this before. We're that little red portion right there in the Great Lakes. This is our main campus and our health science campus. Very nice campuses to work at. So the question is, why do we want to use zebrafish? And I've tried to start to elucidate that in my last little talk there. These are zebrafish. We're talking about an animal that's probably 1 to 2 inches long, a tropical fish. And where are they found? Well, in the wild, they're found in India and in many places in India. And they are usually found in slow-moving streams and in ponds. Okay? So in this is the last census in these little red spots here. These were all areas that they netted the zebrafish out in 2012. So, again, why do we use zebrafish? Well, zebrafish, for the longest period of time, were used simply as a developmental tool to study the process of development and how an animal develops. And this down here can actually show you, if you can see this little square down at the bottom, you can see an animal developing from a single cell all the way through an animal that looks very much like a fish, because it is a fish, in 75 hours, time-lapse photography. And this is the reason why they were used in the first place. I chose to use my animals a little differently. And that is to look at the phylogenetic tree here and see that they're actually 70% homologous to humans at the genetic level, and they're actually more homologous at the protein level, because many, if not all, of the proteins that are present in human disease are also found as orthologs in the zebrafish. This is simply a very simple developmental timeline. So, zebrafish are what I call in perpetual spring. We can have these animals actually breeding in perpetuity for every week. And these are animals that are actually breeding up here, and you can see the eggs coming out. And they drop through these barriers here that will keep them from being this is what they look like in the wild. Males are slightly more yellowish, and the females are a little bit more bluish gray. And the amount of embryos that you can use or produce is really tied to the amount of animals that you have. I currently have a capability of approximately 2,000 animals in a space that would take up probably the first 3 rows here. So the amount of animals that can be housed in very small spaces leads to the ability for you to actually drive costs down for that next arm of your drug development. Here's a good example of them having clinical relevance, however, and this is from a Nature paper, Nature Medicine, and it was a patient that had a specific mutation. I'm not going to go into the mutation, but it produced lymphedema. Lymphedema is really a collection of more fluid in the body than you can handle, and it goes into the lungs eventually. And there was no cure that they could think of. So what they did is they took this DNA from this individual, and they stuck it in zebrafish eggs, produced an animal model that is produced right here. And literally within 12 months had screened that animal model, found a drug that was already FDA approved for something else, and the patient was basically on that drug and was stable and had lymphatic vessel remodeling going on. This is within 12 months of taking the DNA out of the patient, okay? You can't do that with mice, all right? So this shows you the power of the model. Another reason why you would want to use it, and NIH is taking a big look at zebrafish research and medical funding. This is all of the different areas that will be funding zebrafish research just in the last 5 years. And it's nearly $600 million. Okay? This is our core facility at the University of Toledo. And this is for housing, and this is for behavioral research. We look at, obviously, toxicity and other, but we also look at behavior, both at the larval level, and you can see those little guys actually moving around. This is in a 96-well plate. We have a tracking system that can actually track their movement, how far they go, and for how long a period of time. This is what is called a novel tank test, and a novel tank test is actually something that will test for anxiety. When a person is anxious, what do? They stick to the side, they try and fade into the background. What a fish does is they actually get put into the tank and they dive to the bottom. That's where it's safe. And then it starts to explore. So to look at angiolytics or something of this nature, we can then look at it this way. More on that later. All right, this is our core team. Dr. Schiefer is a chemist. He's a good chemist for, especially when it comes to neurological drugs. I am the zebrafish core facility manager, and the person who does most, if not all of the work of maintaining our facility is a former student of mine, Dr. Wiesner. We have a few things in the works as far as husbandry expansion. We were able to get approximately 5 more of these things as well. Other future things for doing genetic work, especially, but also for injecting, for instance, cancer cells into a zebrafish. Now, you wouldn't think that you could do that because of the immune system. But if you do this between 2 and 3 days post-fertilization, the immune system has not been developed yet. We have a xenograft model that has actually been used in our hands. So we can then grow the tumor, treat the tumor with new drugs. These are a few of our zebrafish strains. One that was mentioned in one of the articles that was recently published was this Casper strain. This is a strain that has 2 mutations that literally gets rid of all of its coloration. You can see everything inside of it. It's translucent. And the red, that's its blood. And you can see the blood going through everything. But different ones will do different things. This is one example. This is FLE1, which is a marker for the cardiovascular system. This is literally lighting up under UV the entire cardiovascular system. What we would use this for is to study the cardiovascular system, sure, but we could also use this to look at cancer and angiogenesis. So you could actually take a cancer cell, have it in one particular color, and then have this one in green, and you could see where the two come together, and you could study whether your compounds were actually causing the lack of angiogenesis. Okay? Another one here, this is EGFP on NF-kappa B, and what we're looking at here are neuromast cells and some other organs, and we're looking at it's really a cool thing to look at under the UV light. Okay, so use of the zebrafish animal model at UT, and this has just been over the last 5-6 years. This is Dr. Schieffer. We have a couple of grants here, including a new UO one that was just given to us last year with Dr. Hall, myself, and Dr. Schieffer and a few colleagues, including those at NIDA, for about $2.6 million. This particular picture here is the cover of AC Chemical Neuroscience, and it shows everything that happened for the entirety of this particular drug discovery platform. This is basically, we're looking at the in vivo photoaffinity labeling. We're not trying to do this in cell culture. We're not trying to do this in a beaker. We're doing this in an animal. Okay, so how do we do it? Well, as I said, the Casper zebrafish does not have any color. You can't have a lot of color, particularly melanophores, if you are going to try and hit this with UV light. So first we had to develop, and Dr. Schieffer's lab did this, develop a chemical that looked like methamphetamine but was able to do this photoaffinity labeling, as well as somebody mentioned earlier, click chemistry. And you can use the click chemistry to then attach a fluor to be able to pick up any proteins that might have bound to your chemical. Okay? So what we did first is we did the actual experiments by doing some exposure. Then we do what is called the novel tank test. Now, we were talking about methamphetamine. Methamphetamine will make an animal more anxious. Okay? So what we're looking at is, over here, this is a novel tank test, and you're looking at a midline of about the middle of that tank. And what you're looking at down here is actually our tracking system. We have a tracking system that will be able to look at the animal, track it within 20 millisecond delay, and feed all of that X, Y, Z coordinates into the computer so that we have all of that data that we can then do the analysis. And that's what you're looking at here. What you're looking at now is this particular zebrafish and its track down here. And this is all data that is then fed into the computer that we can use for analysis for the behavior. We're not talking about small amounts of data here. We're talking about very, very large data sets. All right. And then what you have to do, once you've done the exposure, once you've got the behavior from that exposure in the same animal, okay, this is important, in the same animal, now you have to do that methamphetamine being bound to whatever the binding partner is. And that's done with the UV light. So the UV light is turned on, and then we get that piece, that protein, actually adhered to by that protein, and by that drug, because it's a probe that can do that. After that, we have, as I have down here, you have to isolate the brain, you have to then purify the proteins, you then do the click chemistry, you put the floor on, and then you run your gels. And the gels are right here. And you can then see with cold methamphetamine, and that is non-fluor, and can you compete away that? And the answer is we found quite a few proteins. We isolated those proteins. We sent them off for N-terminal analysis of what are these proteins. And then we did the proteomics and metabolome type of work. And they identified several different types of proteins that were in that particular grouping. We have Dr. Tillore, and I think Dr. Tillore showed a few other newer things that we've done with him, but our work with cancer researchers in general has been more on the toxicity level of what their compounds have, okay? And he said, indeed, these are not very toxic, and you can see this one was down to 10 micromolar without any toxicity. And this one was also not very toxic even at 10 micromolar. This is another cancer chemist that we have, Dr. Eisenman, and she was looking at brain cancer, okay? Glioblastoma, and whether or not these endiaformin type of compounds should be antagonists or agonists in order to be non-toxic to the animal but still deal with this particular thing. And this SMIF-H2 was an antagonist. Now the problem with this, and one thing that you have to realize, is that there's many ways of actually looking at toxicity. What you're seeing here is in the control animal up at the top, right in this area here, you can see something moving, yes? That's the blood. It's under the microscope, yes. That's the blood. And what you can do is you can actually look at blood flow through the primary artery and the caudal vein, and assess that these controls, the controls up at the top there, are doing very well. If you start adding SMIF-H2, we then started to see a gradation of slowing the blood flow down. If you look over at the right-hand side, at the bottom, you can see that there's almost no blood flow at 10 micromolar. These were toxic. The animals were starting to die, and they had very much degradation in both the face and at the tail. And this was shown also because we can actually assess heart rate very easily. You see at the top graph there that all of the antagonists are actually slowing the heart rate, where the agonists were not. They're almost identical. Okay, and I'm not going to talk about these, but these are student posters I believe they're at. Are they here? Okay. So These are student posters from my lab, and we'll just go through them very quickly. This is actually one to Chu, Austin Horton, and this is the methamphetamine project. But Tua actually did something that we hadn't seen before, which is to quantitate. Can you quantitate the methamphetamine that gets in? And indeed she did. Another collaborator that I have is very interested in M1 receptors, muscarinic M1 receptors, and particularly repetitive behaviors. And this is a cancer, a chemical design poster. The reason I have that one in there is because you then have to do the pharmacology to say it does what it's supposed to do, right? And then there's the animal behavior poster, and those will all be out there as well. And then this last one was my last graduate student to graduate, Kavita Guacha. And she looked at, and I think she did a very good job, doing pharmacokinetics in zebrafish. So not only does it get in, but tracking how much gets out and how fast. This is another one of my students who's now a PhD student in our College of Medicine. And she's looking at the effect of short-chain fatty acid butyrates on cardiovascular parameters. Now, this slide was put together before I came here. So this was something that we were working on for a long period of time, but we just got word before I came over here that this is probably it. In fact, we've got just in time. They asked for all of the proper things to fund the grant. But this is novel anti-opioid screening. And fentanyl was mentioned earlier. And this is the tracking system, and this red is movement by the animal. Fentanyl, what happens with fentanyl? You slow everything down. Yes? Okay, so how we've actually tracked it is from vehicle to 0.3 micromolar fentanyl, 3 micromolar fentanyl, 30 micromolar fentanyl. And you can see a very quick gradation of inactivity due to the drug. Now, what does that look like? Take a look at this up here. In the light, they have very little activity. And as soon as that hits dark, you see the activity. So they're very much like mice in that they become much more active as soon as the dark hits. As soon as the light comes on, they slow down again, okay? So what were we able to show? This is the control. This is the vehicle. A lot of activity. 1 micromolar, 10 micromolar, 100 micromolar fentanyl. A very good gradation of no activity to no activity. Okay, and we showed a couple of curves for total distance and fentanyl. We get to a certain point, it was very, very difficult for us to do any rescue here, because what we wanted to do eventually was to produce something that did rescue. So if you go to this slide here, you saw a lot of activity with vehicle, but fentanyl, no. This one was with fentanyl, and then with fentanyl and naloxone. So fentanyl was down here, fentanyl was at the bottom, fentanyl with and naloxone, it rescued. 1 micromolar to 1 micromolar. And then you can see how that kind of does fine until you get to a certain concentration. You get to a certain concentration out there, you can't rescue it. Okay, that's enough. Thank you for your time.
Now, thank you, Dr. Frederick, for such a nice information on the zebrafish. Now, session is open for the discussion. I invite only, say, few questions, or no 2 questions, because we are running short of time.
Sir, I have a question regarding that you show that opioid and that brain delivery, especially so when we are in brain we are considering blood-brain barrier and toxicity you are studying, sir. So, and zebrafish.
So how does it get in?
Blood-brain barrier, yes, sir.
Okay, there's different pharmacokinetics from them than from an adult. A larval animal is more like a sponge. And it goes through the skin, it goes through the eyes, it goes through the, obviously the mouth and the gills, just like you would be swallowing something as well. But, so it reaches quick. The concentration that's going to get in is going to get in very quickly. So I guess that is a lot different than what you would see in an adult. And we're going to be doing the adult studies eventually as well.
So I just really wanted to know whether it is based on quantitative measurement or just you have seen like because like in 0.1 micromolar dose and 0.1 and 1 micromolar dose in both the doses the blood flow was almost similar.
Yes.
So,
yes.
And
So what you're seeing is that you get to a certain, and there's a drop-off. There's a major drop-off. And to answer your first question, it's pretty empirical as far as that goes, as whether or not the blood flow is, there's no numbers that we can really put on it. There is simply an observational method, and it's pretty empirical as to what, how you call that. But I think that's rather dramatic, so it's not hard to actually tell.
And my second question is, can we study the amyloid beta plaques, ICV injection amyloid beta plaque studies for Alzheimer's model that we do in mice and
Okay, I'm going to send you to approximately 5 journal articles from my lab on a chemically induced Alzheimer's disease model in the zebrafish. And yes, we've been able to identify not only plaques but tangles.
Okay.
And we actually had a couple of compounds that when they were put into the exposure profile, they eliminated a lot of those issues.
And is there any tau protein model available for zebrafish?
Yes, there is.
Okay.
There is.
Thank you. Thank you so much. Thank you.
Thank you, sir. Thank you so much for your words of wisdom. It will be a great pleasure for us to listen to you again in our future events. Now, I would request Dr. Rizwan Khan, Principal SKICON, to present the memento to Dr. S.K. Jain and Dr. V.K. Dikshit, the session chair, and Dr. Frederick E. Willem, the keynote speaker.
I think time is running. I can only understand.
लगेगा तो हम लोग